talk about ion exchange chromatography. So basically what's happening here is we're going to be separating out different proteins based on their charges. So we can use a positively charged bead like DEAE if we're trying to get out a negatively charged protein. So these beads are going to be located in this here, which is called the stationary phase. Basically, we're going to run a buffer through here, and gravity is just going to pull the buffer through, and anything that is the same charge as our beads are just going to elute out automatically. So if we were to run a positively charged and negatively charged proteins through this, and we had a positively charged bead, like DEAE, well, our positively charged protein is going to elute really, really fast because it's not going to stick to the positively charged beads because the same charge is going to repel each other, right? So, and just some naming stuff. When we say anion exchange column, that means that we're going to be using cationic beads and that we're getting the negative protein by using the positive bead. So similarly, if it's cation exchange column, we're going to be using negative beads. So this kind of shows what it'll look like at the beginning, and then we'll run our proteins through, and at the end, we'll end up with our bound proteins at the very top, and our cationic proteins at the bottom, which are going to elute fast. And then after that's done, we're going to want to remove these tightly bound proteins. And the way that we can do that is by adding a high salt concentration. So the reason that that's going to make the proteins leave that are currently bound is when we add tons of salt, salt has both cations and anions. So if we had our bound protein were anions, let's say, or negatively charged, then chloride ions are actually going to come in and flow, or the whatever anionic species is that the salt is using, and it's going to take the place of each of the current bound proteins to the beads. So it's going to kick off all the bound proteins from the beads, and you'll see that these proteins will be displaced because they're competing for those electrostatic interactions. So then at the end, we'll end up with our protein. Another way we could do that is also by changing the pH. So yeah, we'll use ion exchange chromatography if we're trying to separate out proteins with similar charge or with opposite charges.